Very good afternoon to one and all. Today, we are here with Dr. Professor Sakashi Sato from the University of EE.UU, Department of Civil and Environmental Engineer, and he is an environmental engineer and science educator originated from Japan, and right now he is a US citizen. For the past 3 decades, he has been faculty member of IDAHO State University, where he serves as director of PhD program in engineering and applied science and the MS program in environmental science and management. And also Dr. Sato hold an MS in environmental health engineering from University of Kansas and PhD in environmental engineering from University of Iowa. Dr. Sato has been recognized with several prestigious fellowships throughout his career. In 2002, he was awarded a Japan Society for Promotion of Science. It's a well-known fellowship. It's called as JSPS Fellowship. During this period, he conducted his research work at the Research Institute in Tsukuba in Japan. In 2012, he received a Fulbright Fellowship, and he had a chance to visit the University of Tribhuvan University at Nepal. In 2014, he was an invited visiting professor at Kyoto University, Graduate School of Engineering, Research Center for Environmental and Quality Management. Additionally, in 2017, Dr. Sato was honored with the Chinese Academy of Science President's International Fellowship Initiative. And also Dr. Sato has made a significant contribution to the field of scientific writing, publishing the Handbook for Scientific English Writing in 2009, a widely used resource in Japan. He has served on the editorial board of various academic journals and is currently a guest editor for the special issue of Microbial Fuel Cell, third edition in the Journal of Energy. So today I invited Professor Sato to deliver his guest talk in our Cicata-Altamira. And hereafter, the floor is yours, Doctor. Okay.
For such a kind introduction, my name is SATO. I'm supposed to say something in the Buenos Studies. Good afternoon. It is my great pleasure. It is truly an honor to be here with you today as a speaker at this prestigious institution. I'd like to express my sincere gratitude to the organizers for asking me to come over to present my research work. And thank you all for your presence. So the title of my talk today is Integrated Microbial Fuel Cell and the algae cultivator system. And subtopic is effect of ceramic membrane ion permeability on performance. So this is the outline of my talk today, starting from introduction with brief background and also research goals and objectives. Then move on to microbial fuel cell. Oops, sorry. And the algae cultivator system, experimental method, result discussion and conclusions, and finally, I'd like to address to any questions you may have. So what we're observing right now as a trend, the global population keep increasing exponentially. This is the population here. This is exponential growth. And we expect the population reach 8 billion by 2050. We see industrialization in all countries. The cities become larger and larger. And most important, probably we see global climate change, which is drier region, much drier, and wet region, it's getting wetter and experiencing flooding. So what you see here, trend will be, we need more energy. We need more fresh water and food. We need to handle the solid waste and need to treat wastewater. What the challenges we have today is we need energy, more energy, which is just energy, we need clean, renewable energy. Then energy has to be transported, in this case, the transmission line. This happened to be drinking water treatment plant, one of the largest, which is Osaka, Japan. We need new type of food production, vertical agriculture. And this is Pocatello, Idaho for wastewater treatment plant. That's the idea. The water needs to be treated and reused or recycled. So the goal of this study, I'm just presenting, is we have a general goal and a specific goal. The general goal is to develop sustainable energy from wastewater system which is capable of simultaneously generating electricity, treat wastewater, and produce some value-added products using wastewater as renewable source of energy and other materials, such as nutrients. The specific goal is to develop microbial fuel cell combined with algae cultivator system to recover energy from wastewater as electric power. Then treat wastewater, meaning remove organics in terms of COD and nutrients, then produce algae as bioenergy or biofuel, recovering nutrients from wastewater. So biofuels are low carbon footprint energy. Therefore, much attention attracted to this study. And then we created, so let's do the energy, So this study led to the different generation of energy studies. The generation 1, generation 2, 3, and 4. So briefly, the first generation, they use food, the edible foods such as corn. Such as corn or soy or sugarcane. The advantage of using this material is the process is simplified. So requires less process steps. The challenges or disadvantage is the debate of the created, sparked the debate, which is more important. Food is more important, or the energy is more important because you are taking food converting to energy. So now becomes second generation. Then we use non-food plant biomass, such as straw, wood, or grass. Advantage is now you alleviated fuel versus food debate, but now because of this material, it need more steps to produce fuel. So cost getting higher. Then third generation research use algae. The algae is much easier to process compared to like straws and wood. So requires less process steps, which reduces cost. Then the alleviates the debate between either fuel or food. But still compares to the fossil fuels, the cost is still expensive. The fourth generation use biotechnology, genetic engineering, or carbon capturing technologies. This supposed to reduce the cost. However, this study is still underway. Right? And also, once you generated the DNA modified genetic materials or microbes, or once you release it into the environment, we don't know what's going to happen. So our research focusing on the third generation, which is algae. So algae contains the lipid varying from 20 to 80%, depending on the species you choose. They have a carbohydrate, which converts to ethanol. We have a cellulose, which converts to biodiesel. They have a protein, which can be collected or harnessed as a feedstock. So they have the advantage of using algae. So here, let's compare the advantages and disadvantages. RG grow very fast. In the lab, the 1 week, we have 10 times more biomass. So high production rate. And cultivation does not require land, which reduces competition with land-based agriculture. There is no need for land. And you can use wastewater because the nutrients. Right? You don't need fertilizer. We use just wastewater, which is renewable. So using wastewater as a nutrient source, which reduces cost. Algae lacks lignin, which means less steps for production of biofuel, which reduces the cost. But there is disadvantage using algae. Require large amount of water. Now, but we can use wastewater, and then you can. Other issue is when you use wastewater and controlling the environment, trying to get the optimum growth of algae become more difficult. And because your algae is a product, you are exposing to wastewater. After that, you have to clean, right? So cleaning cost increases. So these are the major disadvantages. So in order to produce algae, there are 3 steps: cultivation, which is growth, the pre-treatment. This needs to be one of the most important part of the cost. Then your production, refinery, extracting, lipid, and purification. So challenges is pre-treatment. Again, once you use wastewater, it reduces cost of nutrient, but increases the cost for the pre-treatment. Okay. So this is the most challenging part of this process is the cost for pre-treatment. So our research objective is to develop microbial fuel cell combined with algae cultivator system to separate so that we don't mix wastewater and algae. So produce clean algae, which reduces pre-treatment cost. And the other objective is to identify growth limiting factors. What can I increase? In order to increase algae growth, it's to optimize. Some parameter is always limited. Right? So identify parameters so that we increase the growth of algae. This is not microbial fuel cell. Just trying to show how the fuel cell works. This is a hydrogen fuel cell. But the principle is the same. So we need anode and we need a cathode. Then between anode and the cathode, we need to separate, which is PEM, proton exchange membrane. So in this case, hydrogen is a fuel. Our case, wastewater is a fuel. Right? But the wastewater organics have the hydrogen, carbon, and oxygen. Right? So here, hydrogen is used right here. Hydrogen is converted to proton and release electrons. Right? Proton migrate from anodes to cathode. Electron flow through external circuit to the cathode. Then electron acceptor is needed. In this case, oxygen is cheap and freely available in air. The oxygen used as the electron acceptor. We have oxygen, the proton, and the electron combined. The final product is water. The microbial fuel cell is the same, and the product is water. So there's no harm to the environment. And microbial fuel cell, we use bacteria as a biocatalyst. So at the anode, the bacteria degrade organics to release proton and electron, and carbon is converted to carbon dioxide. So simply describe for the microbial fuel cell, there are many different kinds of microbial fuels. Dr. Satish has one different kind of microbial fuels. I'm not discussing his kind. So, all right. So basically, you can have a single chamber and dual chamber or two chamber microbial fuel cell. Other classification will be mediator assisted microbial fuel cell or mediator-less microbial fuel cell. Then we have the mixed culture or pure culture microbial fuel cells. Right? So here we have this is the dual chamber microbial fuel cell. Just like a hydrogen fuel cell, we need anode, we need a cathode. Then we have a separator. This is a proton exchange membrane. This is the fuel is introduced, in this case, wastewater. This is the bacteria attached on the surface of anode. The bacteria decompose organics, release proton. Proton migrate from anode to cathode. Electron transfer to anode, flow through external circuit to the cathode. The electron acceptor, we use oxygen. The oxygen takes electron and hydrogen to form water. Okay. The carbon becomes carbon dioxide. This is two-chamber microbial fuel cell. This is single-chamber microbial fuel cell. Okay. The anode chamber is the same. This is the cathode. Electron flows through external circuit to the cathode. There is no the cathode chamber because we remove all the cathode chamber. This is called the air cathode. In other words, the cathode exposed to air. So here, the oxygen in the atmosphere, air, takes proton and electron to form water. Okay. Each system has advantage and disadvantage, but I'm not talking about it here today. Then we have a mediator-less and the mediator-assisted microbial fuel cell. So this fuel cell, this is the anode. The bacteria, this is bacteria. This bacteria able to transfer electrons directly, right? And this is anode. This is the bacteria. This bacteria electron has to be transferred through mediator. So this is mediator and electron transfer to the anode. There are a number of studies on the mediators. Dye will do it. Even the humic acid can serve as mediators. Now, our study, we simplify, we use single chamber and also mediator-less, trying to reduce the cost or simplify the process. This is simple. This is the one of the first microbial fuel cell we use. This is single chamber, very simple. This is the one we use to the research I'm presenting today. Oops. Okay. So this fuel cell has 4 plates of, this is the bamboo charcoal. we try to use as natural material as possible. This is the bamboo charcoal, which has a very low resistance. And for anode is connected. So bacteria oxidize organic matter, release proton, electron go flow through the external circuit to the cathode. This is the air cathode. So oxygen in atmosphere takes electron and proton to form water. This is the real picture right here. There's the opening right here because this is the air cathode. The cathode has to be exposed to the atmosphere. So we use algae, it's Chlorella vulgaris, which has been studied. Many people use this algae because of relatively high lipid content. Dr. Wu published, they say about 14 to 22% lipid. Jablonska reported as high as 56% of the lipid content. The briefly experimental conditions, we use room temperature. This is the size of microbial fuels, about 500 milliliter. And the algae cultivator, 2,500 milliliter. Depth 6 centimeters, temperature 22, PAR, photosynthetically active radiation, which is about 65 micromole per square meter per second. Flow scheme is batch experiment. We run 3 units in parallel. These are the fuel, which is wastewater we created. IDAHO is well known for potatoes. We have millions of gallons of wastewater from potato processing plant. So obviously, we use potato wastewater. pH is 6.9, about close to 7. The conductivity is very high COD, 2,670 milligram per liter of COD. You have ammonium, nitrate, phosphate, potassium, high sodium also. Magnesium, aluminum, sulfur, calcium, manganese, copper, zinc. There are more, very small amount. So this is a concept. So this is not the scale. I will have a picture to show. This is exaggerated. This is the microbial fuel cell. This is the anode. The bacteria attached on the surface of anode. So here, this is potato wastewater, degraded by bacteria, release proton, electron transfer to the anode, carbon becomes carbon dioxide. Right? And because of the wastewater has the phosphate, ammonia, and carbon dioxide become carbonate and bicarbonate. This flows through to, this is a ceramic membrane separating wastewater and clean. This is the clean water. That's where the algae is harvested. Right? The electron goes through external circuit to the cathode, right? Screen out all the waste materials by the ceramic membrane. Only ion pass through the ceramic membrane. So we have a bicarbonate, a carbon source of algae. We have a phosphate. We have ammonia. Ammonia becomes nitrate taken by the algae. The The half, this is, notice here, this is half is underwater, half is above water, half of the cathode right here, right? So the reaction takes place in both because the algae produces oxygen with sunlight. The oxygen is electron acceptor, right? And then above half, which is air, which air has oxygen. Oxygen becomes the electron acceptor. So this is experimental, our experimental unit. It's a small lab. We have a 3 unit in parallel. Then we connect it to the data logger. We use LabVIEW, and data will be logged. We do simple calculations, the Ohm's law, right? So we have we measure voltage, and external load we know because we put it in, right? Then we can calculate the current. We know the current calculated, we know the voltage, calculates the power. The power also calculates by voltage squared divided by load. Now power, this is the power density. The power density is the power divided by surface area of the cathode. That we calculate the power density. Let's move on to result and discussion. This is the voltage. So we have a 3 unit. This is the first unit, very messy. And sort of we know why this one is happening because probably we did not clean cathode anode very carefully in the beginning. So cleaning is very important. Cleaning cathode and anode before starting experiment. This is the second unit. This is very stable. This is this is gray. This is the third unit. So the average of voltage is this first one is very low, 0.174 volt. The second one is the highest. This is orange or brown. This is 0.345 volt. The MFC AC3 is a gray line right here, average 0.269 volt. Now, when you look very carefully, this is like a noise, right? But if you expand this portion, I expand this from day 10 to day 15. Day 10 to day 15, I enlarged. You see, this is diurnal fluctuations. Because this operation is 12 minute light, 12 minute dark, right? So it creates the fluctuations, right? This is the day with light, then without light. Okay. But when you look overall, the differences is very, very small. 0.02 volt differences. When you look, this is other people's, this is not my work. This is Ura and Desha published in 2024. When you look at this, this is Tura, this is untreated, the low wastewater. This is treated by wetland. You can see high is as high as like 350, 400. Low is like a 10 volt, like a millivolt. A huge difference, right? So here our process, as I mentioned, half is underwater, depending on the oxygen produced by algae. But the other half, it's not depending on algae. So day and night, it's light or with light. It's both functioning, both, right? So the previous author mentioned that, okay, we can run 24 hours, but with light. With light means energy. Right? Our system run day and night, so we don't need to light during the night. So it saves the cost. Okay, so let's talk about power. So as I mentioned, first one is very messy. Highest is 40 milliwatt per square meter. Right? And the second one is the best one among three, produces 138, about 140 milliwatt per square meter of cathode area. The third one is about 80 milliwatt per square meter. This is the RG. We measure RG based on the suspended solid. We filtered and weight, then divide it by sample volume. That give you the concentration of RG. So I'm not going to detail because we have a limited time. Just to compare, this is unit 1, AC1, right? Compares to 1 and 2. 1 is significantly better growth, right? This is the pH. Right? Again, pH is much larger in first unit, AC1, compared to the other. The pH goes up to 9.8, almost 10, pH 10. When you look at pH 10, PCPH diagram, right? Almost bicarbonate and the carbonate is almost same concentrations. Right? So where the carbonate come from? Right? This is a closed system. Only places carbon come from is a microbial fuel cell. So within microbial fuel cell, organic is converted to carbon dioxide. Carbon dioxide dissolves into the wastewater, pass through the ceramic membrane to enter the algae cultivator. Right? So CO2 is coming, which raises the pH, which means more carbon, which means carbon is available for algae. I cannot move. I have a problem. Okay. Oh, but not moving. Okay, good. Oops, sorry. Could you go back in? This is a large file, lots of pictures. I like pictures, right? So it's a large file, may create the problem. By the way, do you have any questions so far? So point is, in order to grow algae, we need a carbon. Carbon has to be supplied. Oh, okay. Good. As I mentioned, I have a lot of pictures. I'm going backward right now. Okay, So that's the pH, right? The pH is related to alkalinity, right? So this is alkalinity that you see here. Alkalinity is again unit number 2. AC2 is higher than other 2 units. So alkalinity is a carbonate and bicarbonate, right? The carbon source. So now AC2 has large carbon in this unit. So phosphate. Phosphate is a nutrient. We need phosphate. So you can see unit number 2 is much larger than 1 and 3. Right? Now this drop right here, as you see the pH is close to 10. What happens is once you have a pH approaches 10, phosphate is going to precipitate as calcium phosphate. So then the phosphate drops due to precipitation. So this unit has plenty of phosphate. So phosphate is not the limiting factor. Ammonia drops very quickly. This is 2 reasons. One is nitrification. The other is uptake by algae. And drops once it's not increasing, that means ammonia is not diffusing from wastewaters to the algae cultivator. It's not easy. Nitrate, first increase here, this region, this is due to nitrification. Then this region is the diffusion from microbials and uptake is balanced, right? Then drops, which means the algae takes nitrate, but nitrate is not coming from microbial cell to algae cultivator. So now problem is here. This is very low concentration. Here, they're about 0.05 milligram per liter. With this system is nitrate or nitrogen is limiting. Sodium is no, it react, not easy to react, and mobile, moves very easily. Right? So permeates from membrane very easy. So this can be used as a control, how fast the ion can move through the membrane. Right? So here again, unit number 2, much larger sodium concentration, right, compares to other 2 units. So permeability of ceramic membrane we use in the number 2 is much permeable than compared to the other unit. This is potassium. Potassium very similar to sodium. We know that from a periodic table. Again, number 2 is much, the permeability is much larger. Total dissolved phosphorus, right? So again, number 2 is superior. Conductivity is a measure of current, how close to the, moving going backward. So conductivity is a measure of ions. How much ion in the water? Again, number 2 unit is much larger, which you expect because we have a high sodium, high potassium, much ion concentration is much larger in the unit 2. So this is expected. So this summary, So this is alkalinity. Unit number 2 is much larger. This is algae, sorry, this is algae. This is alkalinity, right? So this is potassium. This is phosphorus. This is sodium. You can see this is sort of obvious. The ceramic membrane used in number 2 is much more permeable, right? This is an issue because this system lacking nitrate. When you compare the maximum power density and algae concentration, this is maximum power, this is algae, this is power, this is algae, this is power, this is algae. This is some relationship. Right? Larger the power, more algae, or more algae, maybe larger the power. So here, so we have a photosynthesis and power output. We have a larger photosynthesis, more algae, more oxygen. Oxygen is electron acceptor, improve the cathode efficiency. Right? Now we have a high power. This we have not proven yet. Might be increases nutrient diffusion from microbial fuel cell to the algae cultivator, which increases the nutrients, increases the algae uptake of nutrients, more photosynthesis. But still, this part needs to be studied. So conclusions. So ceramic membrane allows ion to pass through, right? Diffuse from microbial fuel cells to algae cultivator. Okay. Then algae will take the nutrients to grow. The limiting factor is the rate of ion transport, especially nitrogen is limiting in the system. But once we solve this problem, we can enhance the growth of algae. So future research is focusing on the ceramic membrane, which allowing ion to pass through the faster rate, blocking everything else. Right? We don't want a bacteria to pass through the membrane, then we contaminate algae. We try to keep clean algae to reduce preparation costs, right? Others are many people are working on. We improve, optimize the design of microbial fuel cell or construction and or operation costs, such as the type of wastewater used. We use potato wastewater, could be any other, and nutrient balance and bacterial community and algae type and so forth. So I'm on time, I think. So if you any question, I'd like to address your question.
Hi, yes, thank you for the presentation. You mentioned that some of the other works have the limiting factor about the solar light, and yours is not limited by solar light. How is it that they work overnight?
Over time,
No, the algae, how do they keep synthesizing without light?
okay, because our system is a hybrid system. So we have the cathode, half of the cathode is underwater. That's where depending on algae. Right? But half is above water and exposed to the atmosphere, which has plenty of oxygen in the air. So the night, algae stops producing oxygen, but the air, we have plenty of oxygen. So independent of the light.
Okay, yeah, I get it. Thank you so much. And can I ask something else?
Yes, of course.
And what's the material of the ceramic membrane?
Good question. I had a student from Peru, and he brought the clay, and we had a US clay. We have our university had a sort of the center, they produces pottery, right? So we have two types of clay we compared, and the Peruvian clay is much superior. We look at the electron microscope, very fine. Actually, I had a picture, but I deleted before I came because it's too like that's too big. But it's a very fine, the pore. So we use Peruvian clay, and some happen to be this is a good clay.
Okay, so you have to, if you wanted to make your clay better, you have to nanosynthesize it so it has the small grains as those.
I would do, once you made the clay, I mean the ceramics, I would look at the pore. The pore size we have is the largest pore size is 20 micrometer. That's the largest. The rest is much smaller, fine, the pore size.
Thank you so much.
Thank you, Professor, for your talk. I would like to ask, what could be the main characteristics of ceramic membrane to have a higher ion selectivity in the cell? That is the further work you have to do in this research. The main characteristics for the ceramic membrane.
You mean characteristics of ceramic membrane?
Yeah.
Now, we characterize using the energy diffraction X-ray, and which give you content. But this content, I still don't very, I don't know the which content, because like we can measure calcium content, oxygen, iron, right? Right now, only we know what is better in the pore size. Just looking at the pore size, then we can tell, that pore size, it's not always, it's not good. But also small, then the permeability, reduce the permeability, which reduces the rate of nutrient transport. So there were somewhere between there the optimum, which we have not identified yet.
Thank you.
¿Alguien si quiere practicar con tapón? Sí, también practicar. Doctor, si sabe, usted, you know Japanese language.
Yes, of course.
That's what they asked. Show us in Japanese.
I'll be happy to. Yeah.
Bueno, okay. Thank you, Professor. I have a question. Have you considered genetically modify algae that can produce better results?
Sorry.
Yes, genetically modify algae.
Oh, you're talking about the genetically modified algae?
Yes.
Oh, that's not my area. We have not done anything on the genetics. So I cannot answer. It's not my area. I'm trying to avoid more because of the problem, even though you have developed, you may not get the permission to use it because we don't know once it's released environment, we don't know how do they behave. So getting permission to use it will be very difficult, challenge. So we are more practical, use existing algae, right? Then we probably select high lipid content, algae which has a high lipid content or high protein content. Doesn't have to be the biodiesel, could be food, right? Fish food or chicken food or whatever the useful. Right? Just produce more algae, right? And hopefully this algae helps to produce the more electricity. So there will be the positive loop, right? But we have not done anything genetically modified algae.
Thank you, Doctor, for your presentation. I just have one question, and it's why did you use bamboo charcoal anode and not use another bio anode?
The history go back, the Edison, the first electric bulb, they use bamboo. So that is the study. Bamboo probably it's good to use because it's a natural material. And go to Asia, bamboo is everywhere, right? So and also bamboo grows very fast. Like sometimes overnight, it goes to like a 1 foot or 30 centimeters in the overnight. Grow fast, capture carbon dioxide very fast. Then once you use charcoal, you are capturing carbon dioxide. So the natural material, CO2 sequestration, then high conductivity, the low resistance. So that's what we came up with use bamboo.
Okay, can I ask you another question?
Of course.
What's the efficiency of energy captation from the bamboo charcoal anode?
Efficiency. Okay. We measure resistance. We think that the lower the resistance, it's better, right? Because the resistance, if you have a high resistance, you decrease its power. Right? So we measure resistance. We use because it's a natural material, every bamboo is different. So we just use measure the resistance to pick the low ones. The criteria we use less than 50 ohm. So that all the bamboo have to have a resistance less than 50 ohm.
Sorry, Doctor, can I ask another question? Have you ever tried changing your proton exchange membrane with the Nafion membrane?
Nafion 170.
Yeah, have you tried that one?
No. We started with single channel. The reason, again, same reason, this material is not cheap.
Yeah, it's very expensive, but to have a standard and how competitive your clay is the one that you're using in comparison to Nafion. So how you could be presenting that as an advantage because even though it's not as good as Nafion, it's way cheaper and sustainable.
The reason we use clay has the durability, chemical thermal durability, right? And also rigid. And so that's the major reason we use clay. And cheap.
Yeah.
And available anywhere. Right? Anywhere in the world. It's always clay.
Si alguno no hay pregunta, entonces cerramos la sesión. First, I would like to thank Dr. Chikashi Sato for his kind and very promising presentation towards the microbial fuel cell using the algal system. As a token of gift from Cicata-Altamira, I invite Vigen. Voy a presentar una de las. Thanks for coming, Doctor, for your kind.
Thank you. Gracias.
So I'm going to give a certificate for the presentation. I'm going to read it in Spanish, Doctor, because I try to read it in English. National Polytechnic Institute, Centro de la Investigación, Ciencia Aplicada y Tecnología Avanzada, Unidad Altamira, otorga de la presentación reconocimiento al doctor Chikashi Sato por su presentación en Seminario de Divulgación Científica con el PONENCIA, Integrated Microbial Fuel Cell Algal Cultivation System, Effects of Ceramic Membrane, Ion Permeability on Performance. Altamira, Tamaulipas, 14 de mayo 25, la técnica Servicio de la Patria, la servidor de la directora Rogelio Ortega, firmado de la parte de este centro. Y muchísimas gracias, doctor. Bien.
Thank you very much.